Essay: The importance to humans of the control of growth, reproduction and development of organisms, including themselves. In humans, there are more bacterial cells than eukaryotic cells. Thus, it is important for the survival of the human that these bacterial cells do not develop into pathogenic organisms, but it is also important to maintain these high numbers of bacteria, since they provide a function that is essential for the survival of the human. Humans have special adaptations, such as skin, which act as a barrier to the entry of pathogenic bacteria. Mucosal membranes produce mucus to trap pathogens, bacteria, and acid pH in the stomach to destroy pathogens that enter via the mouth. Bacteria are able to live on the surface of the skin, and those living on the surface of the skin are able to outcompete potentially pathogenic bacteria, and in so doing allow the bacteria on the surface to reproduce, protecting the human from pathogenic bacteria that may harm the skin tissue and cause damage. Similarly, bacteria in the gut help with digestion. Loss of bacteria from the gut can lead to the development of pathogenic bacteria such as E. coli, which can cause diarrhea and severe loss of fluids, which in some cases can lead to the death of the human. By ensuring that the gut flora of the bacteria are healthy and non-pathogenic, the risk of death from pathogenic gut bacteria can be reduced. In the case of unwanted growth of pathogenic bacteria in the human body, humans have discovered that antibiotics can be given. These drugs are compounds based on those released by fungi or other bacteria to destroy the cell wall of particular bacteria, and this can help to remove the unwanted bacteria from the human and help to improve their health. However, too much use of antibiotics can lead to the development of resistance to the antibiotic within the bacterial population. This is because the antibiotic acts as selection pressure, leading to the artificial selection of all bacteria that are resistant to the antibiotic. These bacteria survive, and the non-resistant die. By developing drugs that work by targeting other parts of the bacteria's life cycle, it may be possible to develop antibiotics that are able to work against resistant strains of bacteria such as MRSA and C. difficile. Humans can also use bacteria with resistance genes in their plasmids to help in the development of treatments for diabetes. For example, in the field of genetic engineering, bacteria with resistance to particular antibiotics are being used to identify which bacteria have taken up a plasmid containing the gene that, when expressed, causes the bacteria to produce human insulin. This is advantageous as it means that insulin need not to be taken from other humans or non-human sources, but may be developed and produced in a fast and clean manner with very few ethical considerations. The bacterial plasmids are removed and cut with restriction endonucleases. The sticky ends are made and complementary to the sticky ends made using the same restriction enzymes which are used to cut the gene for insulin. The two pieces of DNA from the plasmid and from the insulin gene are then sealed using DNA ligase and heat shocked into the bacterium in order to take up the plasmid. Then the bacteria are grown on petri dishes containing antibiotics. Those bacteria that do not take up the plasmid will die, then make a copy and repeat the process on a different type of antibiotic. Those that die on the second plate are the ones with the inserted gene in the gene for resistance to the second antibiotic. And because they have their antibiotic gene resistance destroyed by the insertion of this insulin gene, those are the ones that you want to isolate in order to reproduce and grow and thus produce insulin. Humans can also grow other organisms for help with medicines and also for food. For example, growth of plants in greenhouses helps to optimize the amount of growth by optimizing the conditions for photosynthesis. In plants, light intensity and carbon dioxide levels can be limiting factors. By removing competing species by the use of selective weed killers and regular weeding, as well as removal of pests such as caterpillars using pesticides, then the energy generated through photosynthesis can be used to help the plant grow and produce fruit or vegetables or seeds. The plant growth needs carbon dioxide since carbon dioxide is fixed in the Calvin cycle by the enzyme ribisco, which binds to the ribulose bisphosphate to form GP, and then ATP and NADPH are used from the light-dependent reaction to form hexose sugars. The more light reaction and carbon dioxide available, the faster the Calvin cycle will go and the greater the levels of hexose sugars will be produced. The light-independent reaction can be optimized by using artificial lighting, ensuring plants are not placed in the shade. Keeping the greenhouse warm and humid will provide optimum conditions for growth by ensuring that the enzymes are working at the fastest, producing more enzyme substrate complexes, and the humidity will help to reduce the water loss by transpiration stream, since it will lower the gradient for the water potential difference between the inside of the leaf and the outside. By keeping cells turgid, it is possible to keep the plant healthy and functioning at the optimum speed for growth. The more growth, the more food produced, the more humans benefit. Humans rely on the growth of plants to feed themselves and other organisms such as cattle. To increase the yield of meat from cows and to ensure that energy input is as near to the yield from each cow as possible, humans can reduce the movement of cattle, keep them warm, provide them with high source of protein for muscle growth so that the yield of meat is high. Humans also harvest the animals as young as possible so that no energy is wasted on aging, only on the development into adult, and once the organism reaches its largest size, it will be culled. The yield of meat can be optimised by treatment for disease prevention, administration of antibiotics and pest control, by selecting the individuals from the population to mate and only allowing those individuals with the highest milk yield or the highest meat yield to interbreed, will also help to ensure that the gene pool from which individuals are being bred is optimised. For yield, cattle can be put out to pasture and in so doing help reduce the growth of plant species such as trees and bushes. This is a type of stalled succession or deflected succession. The continuous grazing by sheep or cattle of a field will prevent the establishment of a typical oak woodland as a climax community. This allows an increase in the biodiversity of species available in the country, since not all habitats are able to reach the final climax community. Instead, they reach a plagioclimax, an artificial climax. Improvement of pasture land can be achieved by increasing the levels of nitrates to the soil, so this can be done by spreading the manure from the cattle themselves, or artificial sources of nitrates can also be added. This can lead to an increased growth rate of grass species due to increased nitrates, which are needed as a source of nitrogen to the plant so that it can be used to generate ATP and amino acids needed for growth by the production of proteins and DNA for mitosis. Mitosis in humans helps with growth of the organisms and is essential in the growth of the baby into adulthood, as well as repair of tissues by the growth of the cells. The cells that grow by mitosis are genetically identical, and this is important for the survival of the organism as part of repair of tissue following damage. In some cases, divided cells can express genes such as oncogenes, which leads to uncontrolled cell division, and this in turn leads to the development of tumours. Some tumours are benign, which means that they are typically encapsulated and do no damage to the surrounding tissue and do not metastasize. However, some tumours are malignant, and these are able to have small parts break away and develop into tumours and other organs, causing damage to the surrounding tissue, and this growth needs to be inhibited to ensure that death does not follow. The development of drug treatments that switch on genes called tumour suppressor genes and understanding of the epigenetic marks and their role in the development of cells that divide uncontrollably helps to develop drugs for treatment. Knowledge of epigenetic marks and the way that the environment may influence their presence is being used to shape the way we eat and behave to ensure healthy growth and development and minimize tumour growth.